This video covers the topic of passive transport of substances across the plasma membrane from Chapter 3 of your textbook.
This screencast was designed to help you achieve the following objectives: explain why the cell may need to use energy to move substances across the plasma membrane, describe primary active transport across the plasma membrane, describe the activity as well as the functions of the sodium-potassium pump, describe secondary active transport, describe uniport, symport, and antiport, relate specificity and saturation to active transport across the plasma membrane. Lastly, describe exocytosis and the various types of endocytosis, including Pinocytosis, phagocytosis, and receptor-mediated endocytosis, and describe transcytosis.
In the previous screencast, we discussed the various passive processes that a cell could utilize to move substances across the plasma membrane. By passive processes, we mean that the cell doesn't utilize any energy or ATP to move substances across the plasma membrane. In this screencast, we discussed the various active processes where substances are moved across the plasma membrane. By active processes, I mean they require that the cell uses energy in the form of ATP. So, why might a cell need to use energy to move substances across the cell membrane? The substances may be simply too large to diffuse across the plasma membrane. The substances may be charged and therefore unable to dissolve in the fat core of the membrane. The substances may need to be moved against their concentration gradient, or from low concentration to high concentration, exactly opposite of the natural tendency of particles to move, and therefore the cell has to expend a little energy. Or it may be that the cell needs to move relatively large quantities of a substance over a very short period of time, and again, therefore needs to utilize some energy in order to do it. All the active processes by which substances are moved across the plasma membrane fall into one of two categories. If the substances are relatively small, like ions or amino acids or monosaccharides, then the substances can be transported actively across the plasma membrane using active transport. On the other hand, if the substances are large, or if the cell needs to move them across the plasma membrane in large quantities, then bulk or vesicular transport is used. Let's first talk about active transport. So active transport utilizes special carrier proteins called pumps to move ions and small molecules like amino acids and monosaccharides across the plasma or cell membrane. Pumps are simply carrier proteins that utilize ATP. ATP energizes the protein carriers and in most cases moves the substances against their concentration gradients, that is, from low concentration to high concentration. This helps to maintain the concentration gradient. To illustrate this, let's look at this figure from your book. The solute is symbolized by the green squares. Notice that the cytoplasm has a higher concentration of the solute compared to the extracellular fluid. So there is a definite gradient between the cytoplasm and the extracellular fluid for this solute. ATP energizes the pump, which is shown in purple, and that allows the pump to move the solute from low concentration to high concentration, and to maintain that concentration gradient. While discussing active transport, I'd like to briefly discuss a very specific pump called the sodium-potassium ATPase pump, or simply the sodium-potassium pump. So important is this pump that it is responsible for about 2/3 of the resting metabolic rate of your body. Now think about that for a moment. As you sit and watch this screencast, about 2/3 of the energy that your body is using is being used to run these pumps in the plasma membranes of all the cells of your body. That's how important this pump is. In the human body, the sodium ion concentration is greater in the extracellular fluid than it is in the intracellular fluid. In this figure, sodium ions are symbolized by the orange triangles. Potassium ion concentration is greater in the intracellular fluid, the cytoplasm, than it is in the extracellular fluid. Potassium ions are indicated by the blue circles. It is the job of this sodium-potassium pump to maintain these concentration gradients. Now let's look at how this pump operates. Sodium ions inside the cell bind to the pump. ATP is cleaved to ADP, releasing energy which energizes the pump and causes it to change shape. This change in shape releases the sodium ions into the extracellular fluid. There, potassium ions bind to the pump. The pump then changes shape again and releases the potassium ions into the cell. The sodium-potassium pump is now ready to bind more sodium ions inside the cell.
The sodium-potassium pump that I just described illustrates what's called primary active transport. That sodium-potassium pump shown here on the left directly used ATP, or energy from ATP, to move sodium up its concentration gradient and potassium up its concentration gradient. However, there is a type of active transport which is called secondary active transport, and it's powered by primary active transport. So here on the right of this figure, we have another carrier protein. And that carrier protein is going to take advantage of the fact that there's high sodium levels in the extracellular fluid relative to the intracellular fluid due to the work of the sodium-potassium pump. This carrier protein will bind sodium, and at the same time, glucose will sort of hitch a ride. And so this carrier protein will move sodium down its concentration gradient and glucose up its concentration gradient along with it. This is referred to as secondary active transport because we are moving a solute, in this case glucose, up its concentration gradient. And we're not using ATP to do that. We're using the sodium gradient. As sodium moves down its concentration gradient, we're using that sodium gradient to power this carrier protein. And that sodium gradient was established by the sodium-potassium pump. So this is secondary active transport. It's not using ATP directly. It's utilizing the potential energy that was created by the gradient in sodium, which is maintained by the sodium-potassium pump. Lastly, we can classify carrier proteins that move multiple solutes at the same time according to the direction in which they move those solutes. Notice that the sodium-potassium pump moves sodium and potassium in opposite directions. This is referred to as an anti-porter system, and we would refer to the carrier protein itself as an anti-porter. Notice that the carrier protein that moves sodium and glucose moves sodium and glucose in the same direction. So we would refer to this as a symporter system, or the carrier protein as a symporter. Sim meaning same. It's moving them in the same direction. So an antiport system moves two or more solutes in the opposite direction. This is sometimes referred to as countertransport, by the way. And this is illustrated again by the sodium-potassium pump. It's moving sodium in one direction and potassium in the opposite direction. Antiport system or countertransport. And again, we refer to the carrier protein itself as an antiporter. Symport is where a carrier protein is moving two or more molecules at the same time in the same direction, and that's illustrated by the sodium glucose transporter moving glucose and sodium from outside the cell to inside the cell. And this is a symport system, sometimes called co-transport system, and we would refer to the protein itself as a symporter. Active transport, like facilitated diffusion, also exhibits saturability. As the concentration of solute increases, there is an increase in rate of transport until some maximum rate of transport is reached as all of the carrier proteins are full or saturated.
Cells may transport small substances across the plasma membrane using active transport, but large substances, or the movement of large quantities, require membrane-bound vesicles. In bulk or vesicular transport, substances are surrounded in vesicles and transported out of the cell in the case of exocytosis, exo meaning out, or transported into the cell by endocytosis, endo meaning in. First, let's discuss exocytosis. Exocytosis is the vesicular transport process by which cells expel material in bulk. Substances synthesized by the cell, like proteins, and cellular waste products, are wrapped in a membrane to form a secretory vesicle. The secretory vesicle then migrates toward the cell membrane, merges with the cell membrane, and dumps its contents outside the cell. As the membrane of the vesicle merges with the cell membrane, components of the cell membrane are replenished. This figure from your book illustrates the process of exocytosis. So here we have a membrane-bound vesicle with some substance inside of it. This vesicle was likely constructed by the Golgi apparatus. The vesicle then migrates to the plasma or cell membrane, where it merges or fuses with the cell membrane. And as it does so, it expels its contents out into the extracellular fluid. Notice that as the vesicle merges with the plasma membrane, its phospholipids and proteins are incorporated into the plasma membrane. Now that we have considered exocytosis, now let's turn our attention to endocytosis. Endocytosis is basically the opposite of exocytosis. Membrane-bound vesicles are used to bring substances in the extracellular fluid into the cytoplasm of the cell. There are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. First, let's consider phagocytosis. Phagocytosis literally means cell eating. Certain cells of the body can detect microbes and worn-out cells and move toward them. Once in contact with them, the cell membrane bulges inward and the objects are brought into the cell in membrane-bound vesicles. Lysosomes merge with those vesicles, dumping their digestive enzymes into the vesicle. The objects contained in the vesicles are digested, usable digestion products diffuse into the cytoplasm, while unusable products are expelled from the cell by exocytosis. Only a few cells of the immune system, such as neutrophils and macrophages, are capable of phagocytosis, and they are therefore known as phagocytes. Let's now discuss pinocytosis. Pinocytosis literally means cell drinking. The process of pinocytosis is identical to phagocytosis except for the types of substances that are being ingested by the cell. In pinocytosis, what is being brought into the cell is basically extracellular fluid and whatever substances happen to be dissolved or mixed in that extracellular fluid. In this figure illustrating pinocytosis, here we have invagination of the plasma membrane. That is the beginning of the formation of a vesicle. Notice that the extracellular fluid and whatever happens to be mixed in the extracellular fluid at that location is being brought into the cell in a vesicle. That vesicle then merges with lysosomes, which dump their digestive enzymes into the vesicle. Those digestive enzymes break down the substances within the vesicle, and usable digestion products are released into the cytoplasm to be used by the cell. Those substances which cannot be used by the cell are exported from the cell by exocytosis. I want to emphasize that the process of pinocytosis is a non-specific process. The cell is not targeting any specific substance. It's simply bringing into the cell the extracellular fluid and whatever substances are mixed in the extracellular fluid at the time. The third process by which substances outside the cell can be ingested by the cell using vesicles is called receptor-mediated endocytosis. If a cell wishes to target a specific substance in the extracellular fluid for ingestion, it can do so using receptor-mediated endocytosis. In this process, receptors on the extracellular face of the plasma membrane bind to specific substances in the extracellular fluid. As the plasma membrane invaginates, a vesicle is formed that contains extracellular fluid, various substances that were dissolved in the extracellular fluid, but a large number of substances that specifically interacted to the receptor. So we have a vesicle that has a large number of whatever substance was specifically targeted by the receptor. From here forward, the process is the same as with phagocytosis and pinocytosis. A lysosome would merge with the vesicle, dump its digestive enzymes, digest the substances in the vesicle. Those usable products would move into the cytoplasm where they could be used by the cell, and those substances that could not be used by the cell would be exported out of the cell by exocytosis.
Endocytosis and exocytosis can also be used to transport substances across a cell, and that process is called transcytosis. In this illustration from your book, we're looking at, I believe this is a capillary. This is the wall of the capillary. So this would be like where the blood is in the lumen of the capillary, and this would be outside the capillary. This is a cell. Substances in the blood can be brought into the cell by endocytosis, and then the vesicle migrate across the cell to the other side where that substance is dumped and exits the cell by exocytosis. So endocytosis on one side, bringing the substance in, and then exocytosis on the other. And that has the net effect of moving a substance across a cell. That is transcytosis. Some locations where this occurs is the capillary walls and the walls of the intestines. You'll see examples of transcytosis in AMP2 when you study the cardiovascular system and the digestive system.
Now let's summarize the active processes by which substances can be moved across the plasma membrane. Remember that all active processes require the use or expenditure of ATP by the cell. Cells can move small substances such as ions, amino acids, and monosaccharides across the plasma membrane using active transport. A pump or carrier protein energized by ATP is involved in active transport. Larger substances or substances that are being moved across the plasma membrane in large quantities require vesicles, and the process called bulk or vesicular transport is used. Bulk or vesicular transport includes exocytosis, where vesicles are used to move substances out of the cell, or endocytosis, where vesicles are used to bring substances into the cell. There are three types of endocytosis. In phagocytosis, or cell eating, vesicles are used to ingest microbes or worn-out cells. In pinocytosis, or cell drinking, vesicles are used to ingest extracellular fluid and whatever substances happen to be mixed in with the extracellular fluid. In receptor-mediated endocytosis, receptors on the extracellular side of the plasma membrane are used by the cell to target specific substances in the extracellular fluid to be ingested through the formation of vesicles. Let me conclude by saying that an excellent way to test your knowledge of the content of the screencast is to sit down with a blank sheet of paper and from memory, without the use of your book or any slides, to sit down and recreate this figure from memory, and then to explain each of the active processes that are shown here, just as I have done. You will learn pretty quickly what you know and what you don't know.
Let's now review the learning objectives of the screencast, explain why the cell may need to use energy to move substances across the plasma membrane, describe primary active transport across the plasma membrane, describe the activity of the sodium-potassium pump, explain the functions of the sodium-potassium pump, describe secondary active transport, describe uniport, symport, and antiport, relate specificity and saturation to active transport across the plasma membrane. Lastly, describe exocytosis and the various types of endocytosis, including pinocytosis, phagocytosis, and receptor-mediated endocytosis, and describe transcytosis.